Collaborators, Pete develops management approaches that balance human interests with biodiversity, protecting biodiversity. He conducts research all over the globe with detailed field studies in New York, the Great Lakes, East Africa, Southeast Asia, and Hawaii to gain general insights and identify global patterns. So a little bit of all sorts, which is really exciting. And the ultimate goal is to achieve sustainable use of freshwater resources that maintains the integrity of rivers and lake ecosystems. In terms of his academic career, really impressive stuff, publishing more than 150 peer-reviewed papers, and has had his work cited over 26,000 times, which is exceptionally impressive. He's supervised a suite of postgrad students and has mentored many more postdoctoral fellows, and serves as and is a current member of several different councils and boards, and through competitive funding grants has since 2020 alone secured more than $3.5 million US. So some impressive stuff. And the topics of the projects have ranged on the topics of these projects include things like climate adaptive stocking, climate change impacts on fish mercury, pilot work on lake eDNA, and most recently a project focused on eDNA in Lake Tanganyika. So with that, Pete, I'll give it over to you. Thank you very much.
Thank you so much for that introduction, which was a spectacular introduction, given that you only have a few hours notice that you were going to be introducing this. So it's wonderful to see you all. It's such a pleasure to be here at SAIAB. You're going to see me wander this way and then try to self-correct because I'm supposed to be here, but I feel like a caged tiger when I'm behind a podium or a webcam. So I've been in this area around Makanda for a month and a half now. Many of you in the room, some of you in the room I've met, many of you I haven't. And we tried to arrange this seminar to come as early in my 2.5-month visit as possible, so that I could self-introduce via the seminar. And then I look forward to meeting many more of you in the weeks ahead. I'm still here for another month. I haven't been in SAIAB all that much lately because of responsibilities back in the US that have kept me hyper busy. But for the next month, I'm really going to be making a concerted effort to be present in the building in the postdoc office, one of Albert's postdoc offices, as much as possible. So please don't be bashful. Come by and introduce yourself, or I may track many of you down in the weeks ahead. So it's been wonderful to be at SAIAB. So I've worked in East Africa since 1998. And as an African-oriented fish biologist, SAIAB is legendary as the premier research institute of the continent. And so it was really wonderful to work with Albert to arrange this Fulbright fellowship that enabled me to bring my family here as well to visit for 2.5 months. So what I want to do in today's talk is to give you 3 vignettes that are going to represent kind of the breadth of things that we do within my research team. You'll hear me emphasize over and over again that this is a team effort. So I'm here as sort of an ambassador or representative of a set of very large teams. So everything I'm going to talk about is attempting to integrate across disciplinary boundaries, different ways of understanding ecosystems, always with an eye toward better management, better outcomes for freshwaters, both rivers and lakes, although I'm just going to talk about lakes today, with respect to fisheries outcomes, water quality, biodiversity conservation. Those are the kinds of things that we think about, and we try to think about them creatively and integratively, bringing together the latest tools with just fundamental sort of ethical rules of what kind of system do we want to end up with from a human interest standpoint, from a nature conservation standpoint. So I'm going to give you 3 vignettes that are sort of along those lines or addressing those themes. And so I'll start with just team thanks. I'm not going to dwell on this slide, but again, many people in my team at Cornell that work in New York State exclusively, and everything I'm going to show you is really the dedication of this class. And then this is the group that a month and a half ago, I was out on Lake Tanganyika for a couple of weeks collecting eDNA samples with this team, Tanzania Fishery Research Institute, the Nature Conservancy, Cornell, and the University of Wyoming all came together to dream up project over the last 2 years, and we finally were able to do it in January. I'll tell you about that as the third vignette. So what we try to do is to address these big changes in the world, and we try to address them with an eye toward outcomes that society, we as scientists, but society at large, is expecting of our freshwater. So we want to have, in terms of fisheries, we want to have high catches, we want to have relatively large and typically old fish, and there's species that we like and species that we don't care about as much. So from a fishery management standpoint, we're trying to manage these natural systems for human use, and we're actually pretty selective about the outcome variables that we care about. And there are many people around the world. This is just a graphic from Robert Arlinghouse's synthesis of recreational fisheries. Lots and lots of people in the world that recreate with fisheries, and of course, far more involved in fisheries for food. I tend to focus more on the fisheries for food side. So as we think about those target outcomes, we have, the fish that we care about, they have to contend with environmental changes. And if you take a step back for a moment and think about both the number of changes that are happening simultaneously to our rivers and lakes, and the ways that all of those changes can interact to affect the fish, it's actually a pretty desperate state of affairs if you're a fish in the modern world. So for example, this is a synthesis of data on lake temperature and lake oxygen concentrations, where the blue line is the historical average. That's from before 2020 or before 1990. And then this is the last 20 years or so. Yeah, so before 2000 and up to 2020. So temperature trends, our lakes are warming dramatically. Oxygen trends, something we talk a lot less about, but it's going to be a feature of today's talk. So here's the zero line, no trend at all. Historically, we were at, in the late 20th century, we were losing oxygen, and in the last 20 years, we've lost oxygen even faster. This is for temperate lakes, but integrated across North America and Western Europe. So these are fundamental changes to these ecosystems, warming up and losing oxygen, particularly at the bottom of lakes. We also have the problem of exotic fish species. So this is a global synthesis of invasions into the world's river basins. And basically anywhere that's colored means that there's a substantial proportion of the fauna, which it doesn't belong there. It's been transported across basin boundaries, often across continental boundaries. Does anybody recognize this fish? That's the bass. So this is the smallmouth bass, Microptera stolomiu. This is a huge problem even in North America. So I'm going to tell you about this guy as a bad guy within the second vignette that I'll present. So just imagine if you're a fish, your fundamental environment is changing, and the species that you interact with are changing rapidly. And it's definitely not fun to have to deal with smallmouth bass if you didn't evolve with them. So as we think of now, thinking about the fish production from a fish production standpoint, all of these threats tend to undercut the reliability of our wild capture fisheries. And so often people in the fisheries community are turning toward aquaculture as a solution. And so as somebody who has focused on natural ecosystems and the wild fishes that they hold, this raises all kinds of questions for me about accelerating those environmental changes and potentially introducing species. So chemical shifts, disease introductions, additional competing or predatory species could all be risks. And in settings where you have both aquaculture and wild capture fisheries, there can even be a conflict between these two sources of fish for people, wherein detrimental spillover effects from aquaculture could affect the productivity and reliability of our capture fisheries. So I ask questions like, how can we do better? So not to shut down aquaculture, but how can we be smarter about aquaculture? And so I think along the lines of risk reward assessments or cost benefit analyses. So that'll be the third vignette today. We'll be talking about Lake Tanganyika from an aquaculture perspective. So the overview for today is basically I'm going to talk about brook trout. So two species that have been planted in South Africa. In the North American context, this one, well, sorry, in the New York State context, this one is good, this one is bad. And then I'll talk about the Tanganyika tilapia, a native species, which is being propagated for cage aquaculture in Lake Tanganyika, and some ways that we're trying to help guide smart aquaculture practices there. So this will be a story of climate resilience. How can we help a species that we want to be resilient to shifts in its environment? This will be a story of how do we control a really unpleasant invasive species, and then how do we do smart aquaculture. Okay, so geographically, I want to introduce you to my region in the United States. So there's our US map. New York is in the northeastern part of the US. So now here's the outline of New York State. We've got the Great Lakes region here. We've got Finger Lakes. I live right here. And then there's this big green blob there. That's the Adirondack Park. And here's a blow up of the Adirondack Park now in blue. And it's encapsulating Yellowstone, Grand Teton, Everglades, and Grand Canyon and Glacier National Parks. So these are really famous giant parks of the United States. And you can fit all of them within the boundaries of the Adirondack Park. So this is the biggest protected area in the continental United States, but it's a state protected area rather than a national protected area. What we mean by protected area in this case is especially interesting. So it was, by charter, it was only protected as of about 1970. It is a huge, huge area. They didn't throw all the people out and put up a fence. So it's a different model of protected area. Rather, it's a working landscape. So they basically put all land use transformation on ice, but they allowed all of the existing settlements, roads, et cetera, to remain in place. And so you can visit this giant part of the landscape quite easily. There are not very many roads, but there are enough that you can get into any corner of it. And yet there's a lot of remote wilderness as well. So it's probably the wildest place in Eastern North America, especially at that spatial scale. So static land cover now for more than 50 years. I'll come back to acidification. Very few people. I mean, the population density by US standards, which are already fairly low population density, by global standards, very, very low population density. It's a complete tourism economy. So the entire place, all the people that live there depend, they're at most 2 steps removed from tourists. What brings people like me there is not only this natural beauty, it's got 11,000 lakes and ponds. So this is one of the world's premier lake districts. Been studied intensively in the program that I inherited when I moved to Cornell for about 75 years. We've been doing fishery research in a small number of these 11,000 lakes. So all of the lakes throughout this, all these 11,000 lakes throughout that giant protected area went through a whole century of acidification. So we weren't regulating smokestack emissions effectively until the 1990s in the United States. And so the deposition of nitrogen and sulfur acids was just going up and up and up from the late 19th century through the late 20th century. So federal regulation, the Clean Air Act amendments of 1990, did finally transform that regulatory structure. Suddenly, the NOx and SOx went way, way down. The acids going into these lakes went way, way down. And so we had reached a point by 1990 where the western half of the Adirondacks, all those lakes had been acidified almost to the point that they were biological deserts. Many of the lakes got crystal clear, no fish, very few invertebrates. And so we're looking now at 30 plus years of recovery from acidification. But the acidification was actually highly variable from lake to lake. So we now have a starting point for the kind of research I'll tell you about that's highly heterogeneous among lakes. Some of them had some natural buffering buffering against acidification within the watershed. But for the most part, this is all sitting on granite with very thin soils and very little capacity to absorb those acids. So a consequence that we're only really grappling with in the last 15 years, recognizing and grappling with, is something that we label as browning. So if you've ever heard of dissolved organic matter, dissolved organic carbon, or I brought my transparent mug of tea, this is dissolved organic carbon. So when you make your coffee, you make your tea, that's dissolved organic carbon. And so this is pictures from a colleague of Bear Pond up in the Adirondacks, very clear water in the 1980s, and today much, much browner. You could say that this is greener. It probably is somewhat greener, but it's also browner with dissolved organic carbon. So that colored dissolved organic matter is coming mostly from the landscape. And I don't have time to explain the biogeochemical pathways that make the connection between acidification, landscape chemistry, and lake browning. But suffice it to say, every lake in the Adirondacks is turning brown at an amazingly rapid pace. And so initially, if we went back, say, to the 19th century, or when the Adirondack Park was officially formed in 1970, even, you would have seen this wide range of different levels of dissolved organic carbon, or I'll call it DOC. Some very tea-stained lakes, some relatively clear water lakes. Every single lake has shifted from wherever it started, it's shifted to the left on this DOC spectrum. So for us as humans, we don't deal with these sorts of issues in a direct way. It might be annoying that our lake turned brown, or maybe we're happy that it's more brown than clear and more fertile than it used to be. But there are huge implications of this very simple seeming change. So take a clear lake and a brown lake, same amount of solar insolation reaching the lake surface. The light penetrates much, much deeper. So this tea staining absorbs light, sunlight, very quickly. And so you're basically putting a curtain over your lake. That's really profound if you're a fish, because your entire food web base is built on how much depth integrated light availability do you have. And so if you now cut the light off just at the surface, it not only traps all the heat at the surface, it also means that your productive zone is cut in half, or maybe even down to a quarter of what it was only a few decades ago in some of these lakes. So what we're focused on is down at the bottom. We can have photosynthesis when we have deep light penetration. Photosynthesis is a source of oxygen. If we cut off the light here, and furthermore superheat this surface so that relative to the cold bottom waters, we have very, very stable stratification. The separation between the warm surface and the cold bottom becomes very, very firm. So even if you have a big windstorm that comes across the top, it won't break up the stratification in a highly brown lake, because you'll have up to 7 or 8 degrees Celsius shift at a line here just below the light penetration level. So you set that up and you hold it now for 4 months. You use up all the oxygen in the bottom waters. And so what we've ended up with is, with browning, something that we hadn't thought about much until less than a decade ago, and really work led by Steve Jane. A lot of what I'm going to say in the next few slides is his work. So we got our DOC spectrum here, and this is the maximum depth where the water is very warm. So when we go to a brown lake, the warm water is only up near the surface. We go to a very clear lake, the warm water can extend far deeper. So we're going from 1 meter boundary for the warm water, which we're calling 20 degrees Celsius. That's a brook trout perspective, up to 6 or 7 or 8 or 9 meters. So a big, big difference in the amount of area, which for a brook trout is thermally stressful if we're above 20 degrees Celsius. So this DOC, and you can see that this is an exponential relationship, so very, very sensitive to browning. Here's our same DOC axis here. This is the minimum depth. This is basically how far off the bottom the low oxygen reaches. And so if you have high DOC, the low oxygen can reach up very nearly to the surface. If you have low DOC, so this very low end of the spectrum, the low oxygen is trapped far below the surface. So to show this in a different way, if you think of this is the habitable zone where you don't have an oxygen problem and you don't have a temperature problem if you're a brook trout. So brook trout are happy in between here. So at low DOC, we've got a lot of habitat. But once that DOC crosses some kind of a threshold, that habitat becomes ever more constricted by the difference between this linear relationship and this exponential relationship. And the way that plays out in the extreme case, Tycho Lake here, is this is the deoxygenated zone as we go from June through October. So we're setting up stratification of the lake in June. So we're separating the surface and the bottom, and that bottom becomes increasingly deoxygenated and a very rapid rise actually from down pretty deep, 8 meters, coming up to just 2 meters across a 1 month time period. And then it stays that way for months. This is the superheated surface. So again, we got this temperature boundary set up with stratification at less than 2 meters down because this is a very, very brown lake. And so the red zone here is too hot. Red zone here is too low in oxygen for a fish. And you can see this is a terrible place to live. This lake kills most of the brook trout we put into it every single year. We find typically 1 or 2 survivors, which is something like a 5% survival level. So we stock this lake because our partners want us to for many decades, and it's really not a fish population. So it's out in this zone. Okay, so that's the setup for, we've got kind of the happy zone and then the increasingly stressful zone with browning. So if we now take a step back, think about the whole Adirondack Park, 11,000 lakes, we actually have a ton of data from the 1980s. I'm going to focus just on this stacked bar. So this is out of 1,400 lakes surveyed between 1984 and 1987. And we've classified these into lakes that maintain plenty of habitat for brook trout, lakes that always have some habitat, typically between 1 and 3 meters, between the bottom deoxygenation and the surface superheating. Lakes where the entire water column gets superheated. So these are basically polymictic lakes. They don't really stratify very well, typically fairly shallow. Or squeezed would be like this, where the deoxygenation and the superheating come together. And so for at least a couple of days, sometimes a couple of months, there's effectively no habitat remaining for the fish. So of this 1,468 lakes, when we take the 1980s data and project with browning rates to present, our assessment is that a quarter of the lakes that were surveyed, which is a very large number of lakes that were assessed, a quarter of those are going to be squeezed. There's going to be no habitat at some point during a typical summer. About half of them are constrained, meaning that there is a layer that persists throughout the year that's more than 20 centimeters thick. A quarter of them are overheated. They're just superheated all the way to the bottom. So this half of the lakes are completely inhospitable for at least a couple of days, sometimes a couple of months. The buffered lakes are almost absent. These are the lakes that are deeper than, say, 15 or 20 meters and maintain a well-oxygenated and cold deep zone year-round. So, but they're a trivial part of the overall landscape. So now let's dig into fishy stuff to ask kind of how well can brook trout tolerate these environmental changes. And I'm going to be, in the interest of trying to give you a bunch of different vignettes from the research team, I'm going to be pretty quick with a lot of very detailed data here. We've done a lot of lab physiology tests with these fish, looking at their thermal tolerances and looking at their oxygen tolerances. And so I'm going to cherry pick some of the premier results. So this is the proportion of feed consumed across a temperature gradient. Basically, the fish lose their will to live at a certain point. So above 23 degrees Celsius, you can put food in the chamber and the fish are totally uninterested in it. Up until that point, they eat all the food that you give them, but it collapses very, very quickly. So they might be able to survive an acute exposure that's higher than 23, but they lose their will to live somewhere in that neighborhood. We've done the oxygen tests as well. So this is a critical oxygen concentration. So we're basically stepping the fish down in oxygen concentration as it consumes the oxygen within the system. And here I'm illustrating 6 different genetic types of brook trout. So one of the solution spaces that we see is to get more creative with the kinds of trout that we're introducing. And that might sound like we're going to create a Frankenfish with CRISPR or something like that, put it into the lake because it's resistant to high temperatures or low oxygen. We're not interested in going that far. Instead, we're trying to mine the natural diversity of brook trout and the ones that are currently available in hatchery systems. So we're trying to be practical about this as well. So there, in New York State, there's 6 different brook trout strains that are used. We're testing all 6. And so in the lab, they vary a little bit in their oxygen tolerances, but the bottom line is if you go below 3.5 milligrams per liter, these trout kick the bucket. So dead trout above, below 3.5 milligrams per liter, they start to get pretty uncomfortable above 21 degrees Celsius and lose the will to live at 23 or so. So what do the fish do in nature? So as a field biologist, I always want to ask this question. Anytime I have lab data, we go back to the field. So we have the privilege of being able to use some of these next generation acoustic tags, where we do an implantation of a tag which is chirping out not just, hi, my name is George, but my name is George and my internal body temperature is 12.7 degrees or 21.3 degrees. And it also chirps out the depth that the fish is at. So we can use those time series of individual fish field behavior to reconstruct what the fish is doing across a season. And then we also put in sensors that tell us exactly what the environment's doing throughout that season. So the fish initially, the gray is all of the reports. This was for a lake where we actually only had, this lake killed a lot of the fish we put into it, and this was some of our pilot work. So you're seeing maybe 12 fish worth of data here. So you can see enormous scatter. So at any given time slice, we do have some fish reporting from the surface and some fish reporting from the bottom. But what you can't really see without the black line is the typical behavior of the fish. So we do see excursions upward and downward, actually some pretty surprising excursions that I won't be able to talk about. But this is the average behavior of the fish. So they're going deeper, deeper, deeper as the surface is warming, then deoxygenation really kicks in. So they can't go any deeper than that. And then the surface starts to cool a little bit. I've got the overlay here. Surface starts to cool a little bit. Deoxygenation stays steady. The fish, as soon as you get that cooling, the fish are bouncing back up toward the surface. So why would they do that? They're currently trapped below the photic zone. There is no new primary production, probably very limited secondary production, even though it's oxic, it's below the photic zone in this lake. So you're essentially starving the fish when they're stuck down here. And as soon as they can get up back up, they race to the surface. They're making forays. You can see they're making occasional, some individuals make occasional forays, but on average, they're just stuck. So we can now look at this across many different lakes. We've done a total of 6 lakes so far. And you can see the lakes are very different, right? So look at just at the deoxygenation. Taylor Pond here was just a steady ramp all the way through the beginning of September. We capped off in Pinch Nose. Pike came all the way up and intersected the overwarm area. And Fort Bisbee, we retained this nice space and it didn't deoxygenate until very late in the season. So here are tracks of the individual fish rather than the bulk averages. And you can see there's a lot of individual variation here. This is overlaid with those 6 strains. So what we've been doing is to put all 6 strains into 14 different lakes. And then we're trying to see which strains do best and how that could help to guide a smarter stocking policy down the road. So I don't have time to dwell on this. I could talk for an hour about the individual variation here. This is all work by Honorary, a brilliant postdoc that's been working with me for the last couple of years. Okay, so this brings us to what do we do about it? And this climate adaptive stocking experiment sponsored by the state of New York is our candidate solution. If we put these 6, well, to try to come up with a good strategy. So we put 6 strains into a whole host of different lakes. Some years are naturally relatively cool, some are relatively warm. So if we do this year after year, and we're doing it in 14 different lakes, then we can see what's the degree of selection imposed by the lake conditions on the fish. What are survival outcomes, growth outcomes, behavioral differences, et cetera. So I'll give you we're just at the end of year 4. So we're starting to get enough results to feel like we're learning something finally. But we're going to close out with a bang this year with a massive recapture effort for all the pit tagged fish that are out in 7 of these lakes. So this is going to be everything we know at the moment. So differential survival, there are big differences among the 6 strains. So you can see we've got 3 big winners. This is a hybrid that's by far, it's like 95% of stocking in New York State. This is Temiskaming domestic hybrid. That's a Canadian strain with a artificially selected domestic strain. So it's a hatchery product from the 1950s before we thought about genetics that was selected for disease resistance in hatcheries. This is a second Canadian strain, and this is one of the 3 native Adirondack strains. You can't find it in the wild anymore. It exists only in hatcheries. So those populations have been bottlenecked for about 50 years, and who knows whether they really look like the Little Tupper Lake population anymore. That's what LT stands for. But we got Little Tupper, Horn Lake, and Windfall Pond are these 3 native strains, and then the hybrid and the 2 purebred Canadian strains that are very widely used for stocking. Big differences in survival, and this is with research angling and gill netting. This is the gill netting alone, which is the majority of our sample size. We see exactly the same pattern. So the genotype does seem to matter. We're going to be unifying that with the behavioral perspective from the acoustic tagged fish to try to figure out why. Growth trajectories, we can basically classify these into slow growing and fast growing. To my chagrin, the slow growing are the native strains, the fast growing are the Canadian strains and the domestic hybrid. We're working through more sophisticated statistics now, but there are significant differences there. And this will be the last plot that I show from this study. So we were really interested in the angler perspective of how catchable are these strains. And that was motivated by angler reports. So back when they first started playing around with the Horn Lake strain in hatcheries, they would stock ponds in the Adirondacks entirely with that strain. And the anglers would say back to the managers, "We're glad you're stocking that pond, but we can't catch a single fish." And they said it over and over and over again. So this became a legendary strain as an uncatchable brook trout. Well, it turns out this is our catchability in a little artificial pond and by a single angler. So we put in 30 fish, 5 individuals of each strain, and kept track of the order in which this guy could catch them, the most skilled angler in fishing in our area. And the very last fish he got were the Horn Lakes. The very first fish he got are these Temiskaming domestic hybrids. So this is, again, 95% of the stocking in New York State. This is the Jersey cow of New York State brook trout hatcheries. One of the native strains was fairly catchable. The second is also reasonably catchable. The Horn Lake proved to be almost uncatchable. So this is plotted against a behavioral metric, the really simplistic one. You throw feed into the tank in the hatchery where we're growing each of these strains, and how aggressively are the fish feeding on it. So basically this TD hybrid, it's a hyper aggressive fish. You can see this even when you walk up to the tank in a hatchery. They're just crazy active. These guys cower on the other side of the tank. You throw the food in, they don't budge. You have to walk away before they'll come and feed. So huge differences within the species, right? Usually we just think about the demography of a species. We don't think about the genotypes that are involved. We don't think about the differences in behavior. It's just a brook trout's a brook trout. So everything that we're finding here says a brook trout's not a brook trout. These are not all commensurate brook trout in the hatchery or in nature from an angler perspective or from an ecological perspective. So we need to get smarter about the way that we use these fish. There's no clock in this room. What time am I at?
11 past.
Okay. I'm going to skip those inferences because I really want to get to Lake Tanganyika at the end. So So now I want to switch gears in the same landscape. And I'm going to do something unusual here. So this is a fish and fisheries talk, and I'm showing you pictures of plants. What the hell? Anyone recognize these plants? Dandelions. So I was crushed, frankly, when I came to SAIAB and I looked at the grass in front of SAIAB. I looked at the grass in front of the Rhodes house that we're fortunate to get to occupy for these few months. And there's dandelions everywhere here. So this is the scourge of our area of New York State. It's a European species, been in North America for I think a couple of centuries now. And a lot of us as homeowners actually go out and remove them from our front yard. Some people are doing it chemically. Some of us, I'm old school, I actually fork them out. So in the spring, just before they get to the seed stage. But if you let them get to seed stage, they really take over the terrestrial system if you're in a yard type setting or a shortgrass prairie even in the US. So for me, everything I'm going to tell you about smallmouth bass and their control actually connects to my personal experience. And then, as I said, I was so struck by seeing dandelions everywhere here in Maconda. So I try to control the dandelions in my yard, and it had never occurred to me that I was actually probably messing with the dandelions, selecting on the dandelions to be good at resisting my primitive efforts to control them. So that's the story I'm going to tell you with smallmouth bass. So smallmouth bass do not belong in Adirondack lakes. If you think of that park boundary is set by elevation, and smallmouth bass never made it above 1,500 feet naturally. But now they're in probably a quarter of the lake's parks, and our eDNA surveys are suggesting that they're actually much more widespread, so actively expanding their range within those 11,000 lakes, even in the last decade or two. Which kind of makes sense. Our lakes are warming. They're a kind of cool to warm water fish. So the lakes are becoming better for smallmouth bass than they are for brook trout or lake trout or other historical target species. So some of us want to do something about that. And this is the classic kind of depiction of stages of invasion of a species. So first they're introduced, then they establish, then they start to spread. And then you get to a point where they're a big enough problem that you want to suppress them. And you want to suppress them in two ways. Locally, you want to try to reduce the population, ideally eradicating it. And then you also want to cut off this range expansion step as well. So how do we do that? The bottom line is a great deal of experience now shows that once you get to this point, containment is almost impossible. Suppression is sort of a never-ending task that rarely works very well. And so here's some conceptual graphics, but they capture some important things. So this is the density of an invader. So if you're in a fully colonized system, you're out at the right end of the spectrum. And this curve here, the y-axis is ecosystem state. So this invasion is changing the ecosystem in a way you don't like. So you're out here, you want to get back to here. And so you can convert this ecosystem impact per unit of invader into basically a derivative. That's what this curve is. So there's a domain here on the invader density gradient where you can actually get a lot of positive environmental change per unit reduction in invader density. There's also a domain which you can kind of infer from the plateau here, where you can keep pushing the invader density down and you're going to get very little ecological return. And so this curve is the effort per invader suppression or control. So once you get to a very small population remaining, it's really murder to try to get those last individuals. When they're all over the place, you can actually remove a lot of them pretty easily. And so the question is, if I take this effort efficiency curve and intersect it with the ecosystem response efficiency curve, how low do I want to push things in order to get my ecosystem to the state that I want it in? So we have been doing that experiment for 25 years in 2 lakes trying to suppress smallmouth bass. And I'm going to tell you both a demographic and a phenotypic and a genomic story here, all in 5 slides. So what we've done is actually 4 different cycles per year. We go out with an electrofishing boat in these 2 lakes, and we don't actually fish the entire shoreline, but we fish representative sites, which we think are the best sites for bass, all around the perimeter of these 2 lakes. They're good sized lakes, they're not huge lakes, a couple of hundred hectares. So we do that in the spring, the fall seasons, also in the early summer, 4 cycles per year. So we remove a lot of fish. We kill every single bass that the shock boat brings up. We, for a small subset, but a decent number of fish, we collect trait data and tissues. And then I'm going to tell you stories about genomic differentiation using this 25-year archive of tissues. So let's start with demography. This is when we started in the year 2000. So here's our catch per minute, our CPUE, before manipulation. And so remember, initially the goal is just to wall up the fish. We don't want smallmouth bass in these lakes. Let's see how low we can push the population size. So were we successful? This is an enormous effort every single year. No, this was like, it's like we're completely wasting our time because actually the average CPUE has gone up. And it went up very shortly after we started. So the irony of that is we got several different methods of estimating annual survival. So in the pre-period, so this is with, actually this is pure genomic results here. So inferred survival. We estimate 60% annual survival. Once we started the removal, and this is now 20 years of samples, we're at 24%. So we've cut the survival rate very, very dramatically. So we're killing a lot of fish. It's a large proportion of the population, about half, that comes out every single year. So from a demographic standpoint, that seems like, okay, we're on the right road. We're killing a lot of fish. We've reduced survival enormously. We've removed a large proportion of the biomass. So which fish are we getting that somehow yields this counterintuitive outcome of ending up with a higher CPUE? And the answer turned out to be very simple. So this is the size of the individual fish. These are frequency distributions from every single year up through 2022. And you see a very stereotyped pattern here. So we had a lot of good-sized fish. So a big chunk of the biomass was contained in a modest but not trivial number of large individuals. Before we started hammering them, we've basically taken the head off of that, off of the fish size distribution, and the tail has become huge. So this is not the way fish populations normally work. You don't have rock solid recruitment every single year. Some fish are pretty stable recruitment. Many fish are very episodic. The entire population is built on every N years, you get a really good year class. These fish are getting a great year class every single year because we've taken out the predator. So there's no cannibalism loop here. The large predatory bass are in no way controlling or competing with the smaller ones. And so it's a great place to be a baby smallmouth bass. The growth rates are phenomenal. We kill a ton of them, but the survival is still pretty good. So basically we moved from a population of big bass, few big bass, to many small bass. This is called the Hydra effect in invasion biology. So you chop off the head and you get 2 where you had 1. And we've got very stable population dynamics. So now let's see what's happening below the hood, so to speak. So we've phenotyped all of these fish over many, many, many years, and we see a very consistent increase in reproductive investment. So this is the GSI for the fish nerds in the audience. So we've got our time series here. We don't have we didn't have this data from the entire time period, but we've got a reference lake that we sampled occasionally. And this is the lake that we hammer the hardest. This is age 3 males. So this is the first year that males are breeding. You go to age 4, our sample sizes are very small because we've killed almost all of them. So these are our first reproductively mature males historically. And you can see that the GSI is diverging. It's not a very impressive slope difference, but that actually represents something something pretty substantial, like a 25% increase in GSI in that removal lake. And the H4 females, first year of reproductive of maturity in the females is much more impressive, the divergence between the reference lake and the control lake. And so again, we're looking at something like a 30 or 40% increase in GSI. So what that means is that females of a particular size are putting a lot more of their energy into eggs, and even males, which typically are not all that responsive in terms of their gonadal size, are boosting the size of the testes quite dramatically during the breeding season. So what else is happening? Turns out they're maturing earlier. So historically, we would never see maturity below H3 or H4 for males and females respectively. This is a genome scan, so a Manhattan plot if you're into genomics. We've got 3 chromosomes that are showing multiple loci that deviate between our before genomic profiles and our 2019 after genetic profiles, so 20 years into the control effort. Chromosome 19 is the one that has many, many, many different SNPs that are suggesting a non-random pattern. So we can now delve into just one of those SNPs. This is the allele frequency at the most responsive of those chromosome 19 SNPs. So we actually have some historical scale samples we were able to profile. And then here's our before and our after data. This was the whole genome scan that was used for the Manhattan plot analysis. Then we took a deep dive at just this one locus. And this is the frequency across a total of 1,200 fish represented here in step function change in the allele frequency. So now when we map that allele state to, so the genotype state to the phenotype, to the age of maturity, this low frequency of that allele is strongly associated with maturing at a greater age, and the high frequency of this allele is associated with maturing early. So the bottom line is about a quarter of the fish are now breeding a year earlier than we ever saw them breed before. So to make a long story short, we created weeds in this system by consistently year after year after year in exactly the same way at exactly the same places that what we thought was a virtue. We were doing the same thing, we're rigorous science. This is going to work one day. And actually, we were cultivating a population that was very highly resistant to our efforts. It ends up having spectacular recruitment via both ecological and evolutionary feedbacks. And now we've got a dandelion that it would be impossible to get rid of these fish. If it ever would have been possible, it's probably no longer possible because they grow so fast. This is the growth rates through time. You can see all of these curves are going up. So every single year class, these are the 4 cohorts in the population. They're growing faster than they used to. So yeah, almost uncontrollable. So the combination of faster growth, earlier, greater reproductive investment, and earlier maturation just means that it's pretty intractable to try to control that population. So what we can learn is that we really have to pay attention to the evolutionary or genetic impacts of our management efforts. And I'm going to leave it at that so I can get to Lake Tanganyika. How many minutes do I have left? 10 minutes. Okay. So I'm going to close out with a final vignette. So I deeply love Lake Tanganyika, working there from a cultural standpoint. Biodiversity is spectacular. This is one of the world's crown jewels in terms of aquatic biodiversity. And unlike many, many, many other biodiversity crown jewels worldwide, this one is still in pretty good shape. It's not pristine by any means. And climate change is walloping the fishery, really changing the nature of the ecosystem dynamics, including the temperature and oxygen patterns that I talked about in these dinky little Adirondack lakes. The same kind of stuff's happening in Lake Tanganyika. So this is a lake which is a huge lake, 30,000 square kilometers. This is Tanzania, Zambia, DRC, and Burundi. incredible diversity of fish in the cichlid family. So you're all familiar with those here in South Africa. But you got 250 species of cichlids alone in Lake Tanganyika and about 100 other fish species. And we got interested in eDNA from a biodiversity standpoint, but I'm going to try to tell you a story about aquaculture and how maybe we can be smarter about aquaculture siting based on eDNA findings. So what you need to know as background is Lake Tanganyika has very strong population structure of many, not all, but many of its cichlid fish. And that's because there are sand-loving species, there are rock-loving species, and there are even some species that specialize for a rock-sand matrix. All of this is sand, but above water is rocks. So you get shallow rocky habitats and deep sandy habitats there. So this is the best available, well, remains the best available habitat map. It's actually very wrong, but it captures the right idea, which is extreme patchiness of habitats. So if you imagine, so blue is rock, red is sand, and yellow is sand, and red is the hybrid habitat. So even if this was correct, and actually the resolution of the patching, the grain of the patchiness is much finer than what you see on the map here. But even if this was correct, you can imagine if you really love sand habitat, you have this whole series of discrete habitats with large areas of inhospitable habitat in between. So if you really love sand and you're not going to swim across rocks, which is the case for a modest number of species, if you really love rocks and won't swim across sand, the case for many, many species, then you actually are in an island archipelago. It's one lake, it's wet all around, but you're not going to swim out into the great abyss because you'll get eaten by a lates, an Nile perch type fish. There's 4 endemic lates there. So you can't go out into the open water. You're not going to move along the shoreline because you're in a habitat that you can't function in. So super patchy habitat, which means very fine-grained population structure. And the other thing going on is the lineages are typically 2 to 3 million years old. It's a 12-million-year-old lake. Something happened to keep diversity pretty low for the first 9 million years, and sometime around 3 million years ago, we had this extraordinary explosion of Tanganyikan cichlids, and that's actually coincident with the explosions of most of the other lineages of the lake. So there was early explosion, and that gives rise to really long genetic branches that distinguish different groups of cichlids within the lake. So what I mean by that, this is a long branch as opposed to a short branch. So long branch means that from an eDNA perspective or a genomic perspective, it's very easy to tell these species apart. That's pretty unusual in species-rich landscapes. Even though these are all cichlids, it encompasses most of the cichlid diversity, phylogenetic diversity in all of Africa is contained within Tanganyika. And because the radiations are so old, most of the branches are pretty long. That makes it easy for us to tell them apart genetically. So can we look at population structure using genetics without touching a fish? And can we do it massively in parallel? In eDNA sample, in principle, we could get, there's typically around 100 species, it's believed, at any given location. So in concept, we should be able to, without touching any of those individuals, get population phylogeography for 100 species. So I'm going to cut to the chase here. So we developed a custom ND2 locus, 402 base pairs of sequence. We have a lot of reference data because evolutionary biologists have loved Tanganyika for a long time. Our samples from 2015, it's just 46 sites, had 123 putative species identified by more than 3% sequence divergence. And this is the real kicker, 689 distinct haplotypes. So if you're eDNA speak, those are ASVs. What this means is this is intraspecific variation. 689 over 123 is the average number of unique sequences per species that we have in this 123 species. So on average, you have 5 or more different genotypes per species. It's actually very skewed. Here's just a teaser of 2 extreme cases. So this was just samples from 46 sites in 2 regions. We had to skip everything in between for logistical reasons. But these are 2 important reasons because this is the northern basin and this is a central basin. And there's a saddle, a shallow, meaning 400-meter deep saddle right here. This is 1,400 meters, and this is 1,100 meters. But it comes to only 400 meters there. So historically, when the lake level was down 400 meters, these were two different lakes. So we expect to see vicariance population structure differences between these two regions. So here's just two of the many species. This is Petrochromis famula, a highly specialized rock dweller, very strong spatial pattern. So all the blue sites up at Kigoma are very distinct, and this is 8 or 9 base pairs out of 402 that separate all of these from all of these down in the south. So clear vicariance. This is Lino tilapia dardenai. This guy will swim over any kind of habitat. It can eat almost anything. As a juvenile, it's insectivorous. As an adult, it does a lot more rock algae grazing, and it's just a big mix. So this is a panmixia signal and a strong vicariance signal. So now let's go to aquaculture. That's the endemic tilapia of Lake Tanganyika. So we have this spectacular fish, Oreochromis tanganyiki, really beautiful. It gets almost as big as Nile tilapia. So from an aquaculture standpoint, it doesn't cap out small the way that many Oreochromis do, or tilapians do. And the entire, so at those 46 sites in 2 regions, everybody, 98%, is a single haplotype. There's a couple of satellite haplotypes, very low frequencies. And so we don't have results yet, but in January, we went and sampled 126 sites, the entirety of the Tanzanian coast. We've got all kinds of fun questions about protected area effectiveness, private versus public, different fishery reserves, river inputs, those paleo lakes. We get all 3 of the historical basins represented. But for aquaculture purposes, what we want to know is, given that in the north, all Oreochromis tanganyiki are a single ASV in effect, they're all the same thing. All the tilapia that are used in aquaculture, thankfully, I give enormous credit to the Lake Tanganyika Authority and the fishery commissions of all 4 riparian countries for embracing the Tanganyikan tilapia. They have mandated that you can't grow Nile tilapia or any of the hybrids or the gift tilapia or anything else in the waters of Lake Tanganyika. It has to be Oreochromis tanganyiki. So with this sample set, we will definitely be able to test if there are Niloticus or any of the other tilapians being grown in the lake. We'll also know whether their eDNA is coming in from the watersheds all along this coast. But we sampled the only 6 places where there's currently fish farming. So we now have a baseline for what the genomic, the population genetics, just at ND2, we don't have tissue samples, looks like along this entire coast. Coming back in 10 years after their aquaculture farms all over the place, we should be able to detect have we gotten escapes from the farms? Is there a difference between, well, this will tell us if there's a difference between northern tilapia and southern tilapia. And a key issue is all of the aquaculture tilapia originated with populations in the south. So they're being grown up here and at 6 sites in Tanzania, but they all originally were captured and brought into the fish culture from a southern population about a decade ago. So we're optimistic that we'll actually be able to tell them apart from the wild tilapia, and we'll be able to see where's the break point between the natural distribution of those different strains of tilapia, if you will. So hopefully you're seeing a theme there with the brook trout experiments that we've been doing. So that is everything that I have to share. I think I'm at time or beyond. I would love to take questions, but thanks so much for your attention. If you need to go, by all means. Yes.
Thanks for the talk. It's great to see textbook results every now and then.
Every now and then. There's plenty I didn't show.
The lakes that are changing, going back to your first example, are there any organisms that are thriving under those conditions? Or just a general negative?
That is a great question. So I'm going to try to be brief. So smallmouth bass love the new form of the lake compared to the old form of the lake. And it's really become ideal conditions for them and for some of the other centrarchids that are also here, the Lepomis, the sunfishes. I don't know how many Lepomis have been introduced into South Africa, but at least a couple, I think. So we've got just two that have been, we have one native and two that are introduced in the Adirondack. So any of those centrarchids, the basses and sunfishes, they all love it. Northern pike have been introduced. Thankfully, they have not found their way around as much. Same for yellow perch. So persids and asocids. All these introduced species, they're mostly cool water fish, so they can make it through the rigors of a very long and cold winter, and then they really kick it into high gear much more effectively than the native fish during the short but warm summers. Among the natives, we've I want to just name drop a method. We've used the oxygen isotopes in the otoliths with a fairly primitive analysis, the whole otolith analysis, nothing fancy with lasers. And it's actually really fascinating. We've been able to characterize the entire fauna on a spectrum of growing season temperature using the delta 18O of the carbonate in the otolith. So if anybody's interested in that sort of thing, come talk to me because I'm totally hooked on this method. So that enables us to make some predictions about which species really thrive under relatively warm conditions and which ones absolutely require the year-round cold, including the time when they're growing. So the premier species of the Adirondacks, brook trout and lake trout, they're not happy at all. Brook trout, ironically, were the last survivor among the fish fauna with acidification. They can take acid down to a pH of about 5. Almost every other species was gone by the time the lakes hit a pH of 5 or 4.7. Brook trout can still hang on down to 5 or even into the high 4s. And yet the temperature combined with bass invasions, it's just completely wiping them out. They were the most widespread native sport fish species. There are some of the leucicids, the North American minnows, that do pretty well in warm conditions. So we think that creek chubs are actually going to thrive with the warming lakes. But anything that needs a cold refuge in the summer, its days are numbered. Unless maybe we can help some of them hold on with stocking strategies.
Any questions? Wondering why.
There's lots to think of.
Well, I'm gonna try to make sure that I'm easy to find for the next month here at SAIAB. So I really would love to meet many more of you and hear about what you're doing and compare notes on things we're thinking about.
So this technical thing, the eDNA is depleting passive sampling or active sampling?
So everything I showed was active. We've done some whole lake manipulations now with eDNA, both mitochondrial and nuclear, as targets where we were comparing active and passive. So I couldn't squeeze that in today, but I'd love to tell you more about that. Yeah, so far our passive results actually look almost as good as our active results when we set up a passive sampler at a lake outlet. So essentially the whole lake is very, 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 very slowly flowing past our sampler. In Tanganyika, actually, we attempted to do, we went out there fully outfitted to drag passive samplers behind the boat at 18 kilometers per hour between all of those sites. So we would have the intervening zone, and then we'd have a point sample with active sampling all along every 5 to 6 kilometers of the whole shoreline. And unfortunately, it turns out, at least with the design we had, you cannot drag the thing at 18 kilometers an hour and have it physically survive. So we kept tearing them off or they would fall apart on us, these 3D printed things. So we failed miserably with our passive samplers in Tanganyika. I'd love to learn some tricks.
What one is the active?
My lab is trying to switch to a single platform. Well, we have two key platforms, the Smith root filters. We've done everything now at 5 micron pore size. We switched to the Smith root prepackaged non-desiccating filters for all the Adirondack work, and we're about to sample about 250, 300 lakes in the next 2 years by that method. In Tanganyika and in South America, we're using this awesome self-contained silphium filter kit. They're very expensive on the front end, but in the field, they're so much nicer. It's fully self-contained. You inject a preservation solution at the end, cap it, and it's now shelf stable. You don't have to worry about keeping it cold or anything. 5 micron. You can get a whole range of different pore sizes. They're basically adaptations of dialysis systems.
One final question. Oh, as Pete mentioned, he's going to be around. Please make the opportunity to go and chat to him because you clearly have experience in many different things. So definitely somebody to speak to during his time here. So thank you very much, Pete. Appreciate
Thanks,
it.
everyone.
Thank you. That was awesome.